Today, we are going to begin an important topic in community medicine, Introduction to Biostatistics. This is the first class in the series of biostatistics. To make the subject easier and more interesting for you, the entire course of biostatistics has been divided into conceptual classes. Each class will focus on understanding the basic ideas step by step, rather than rote learning of formulas. Before we understand biostatistics, we must first understand what statistics actually means. So let us begin with a simple question. What is statistics? Statistics is a branch of mathematics that deals with collecting, organizing, analyzing, interpreting, and presenting data. In simple words, statistics helps us make sense of numbers. Whenever we collect data and try to extract meaning from it, we are using statistics. Now let us move one step further. When statistics is applied to biological and health-related data, it is known as biostatistics. Now that we understand what biostatistics is, the next important question is, how do we actually apply biostatistics in practice? In real-life medical and public health work, biostatistics is not used in a random manner. Instead, it follows a systematic and logical process. This process of biostatistics involves a sequence of 5 important steps. These steps include collecting data, organizing data, analyzing data, interpreting data, and finally presenting data. Let us now understand each of these steps one by one in detail. The first step in statistics is collecting data. This refers to gathering raw information from various sources. Data can be collected through surveys and questionnaires, interviews and focus group discussions, direct observations, sensors, or from secondary data sources such as records and reports. While collecting data, it is also important to understand the type of data we are dealing with. Broadly, data can be classified into categorical data and quantitative data. Categorical data, also known as qualitative data, includes nominal and ordinal variables such as gender, blood group, or stages of disease. Quantitative data, also known as numerical data, includes interval and ratio variables such as age, height, weight, or blood pressure. Accurate and appropriate data collection is essential because the quality of analysis ultimately depends on the quality and type of data collected. After collecting data, the next step is organizing data. In this step, the raw data is arranged into a systematic and usable format. Organizing data mainly involves 2 important processes: coding of data and cleaning of data. Coding of data means converting responses into numerical or standardized forms. For example, male and female may be coded as 1 and 2, or yes and no may be coded as 1 and 0. Coding makes data suitable for computer entry and statistical analysis. Cleaning of data refers to checking the data for errors, missing values, or inconsistencies. This includes correcting wrong entries, handling missing data, and removing duplicate records. After coding and cleaning, data is then arranged into tables and spreadsheets. Organizing data in this way reduces complexity, improves accuracy, and prepares the data for analysis. Once data has been collected and properly organized, we then move on to analyzing the data. At this stage, we try to understand what the data is actually telling us by applying different statistical methods. For this purpose, we mainly use 2 types of statistical approaches: descriptive statistics and inferential statistics. Suppose we collect data on ages of patients admitted to a hospital. If we simply calculate the average age or present the data in a table or graph to understand the pattern, we are describing what the data shows. This approach is known as descriptive statistics. So descriptive statistics helps us summarize the data in a simple and understandable way. By using measures like averages, percentages, tables, and graphs, it gives us a clear picture of what is happening in the dataset. Now, suppose we study a sample of patients and use that information to draw conclusions about the entire community. In this case, we are going beyond description and trying to make conclusions about a larger population. This approach is called inferential statistics. Inferential statistics helps us use information from a sample to make reasonable conclusions about a larger population. This is especially useful in medical and public health research, where studying the entire population is often not possible. Now that we understand the 2 main approaches used for analyzing data, let us look at what each of them includes. This diagram helps us visualize the components of descriptive statistics and inferential statistics. Let us begin with descriptive statistics. Descriptive statistics mainly deals with summarizing and describing the data we already have. It includes three important components. The first is measures of central tendency. These measures help us understand the central or typical value around which the data is clustered, such as the mean, median, and mode. The second component is measures of dispersion. These measures describe how spread out or variable the data is around a central value. The third component is distribution measures. These help us understand the shape and pattern of the data distribution, such as whether the data is symmetrical or skewed. Now let us move to inferential statistics. Inferential statistics helps us draw conclusions about a larger population based on sample data. One important component of inferential statistics is hypothesis testing. This helps us decide whether the observed result is statistically significant or has occurred by chance. Another component is regression analysis. Regression analysis helps us study the relationship between variables and understand how one variable changes with another. The final component is confidence intervals. Confidence intervals provide a range within which the true value of a population parameter is likely to lie. Together, these components form the foundation of data analysis in biostatistics and will be discussed in detail in the coming classes. Before we go into further details, it is helpful to first look at the overall structure of descriptive statistics. Rather than studying individual topics in isolation, understanding how these components fit together will make the subject much easier to follow. This diagram gives you an overview of how descriptive statistics is structured. At this stage, it is important to remember that our aim today was only to understand the broad framework, not the details. In the next class, we will begin with measures of central tendency. We will discuss mean, median, and mode, understand what each of them represents, when to use them, and look at simple examples relevant to medical data. Once the concepts are clear, understanding the remaining components, measures of dispersion and distribution will become much easier. For now, just focus on remembering how the topics are connected, rather than memorizing definitions or formulas. This conceptual clarity will help you throughout the biostatistics course.